Thanks everyone to attend this session. My name is Michael Zhou, Marketing Manager for Molecular Biology Portfolio. It is my privilege to introduce speaker for this session, Dr. Jan Joseph Merenhorst. Dr. Merenhorst is Vice Chair of the Center for Immunotherapy and Precision Immuno-Oncology and the founding director of the Cell Therapy and Immunoengineering Program at Cleveland Clinic. Dr. Merenhorst received his PhD from the University of Leiden, Netherlands. In 1998, he moved from the Netherlands to the National Institute of Health in Bethesda, Maryland. In 2012, he was recruited by Dr. Carl Jones to the University of Pennsylvania, where he first served as Deputy Director, Dr. Jones Clinic Manufacturing Facility. In 2013, he was promoted to Director of Product Development and Correlative Sciences. In that role, Dr. Merenhorst was at the cusp of the first ever CAR T cell therapy approved by FDA, Kymriah. The title for today's presentation is CAR T cell therapy, the long-sought cure for cancer. Without any further ado, the floor is yours, Dr. Merenhorst.
All right. Well, thank you very much for this kind introduction and for inviting me to give this presentation on the work that I've done at the University of Pennsylvania with Dr. Carl June. The title, as you can see here, The Long-Sought Cure for Cancer with CAR T-cell Therapies. The work that I'm presenting today is work that was clinically started in the summer of 2010 with the infusion of the first 3 chronic lymphocytic leukemia patients with their own T-cells engineered with a chimeric antigen receptor. In brief, what a CAR does, it actually is made up of an antibody fragment that interacts with a surface protein on target cells and grafted onto T-cell molecules that give it stability, signaling, and endow a T-cell to exert effector and memory functions after infusion. This work started more than 12 years ago, and we've been following these patients for a decade. First reports came out in 2011, and patient number 2, the second patient on this trial, was reported in New England Journal of Medicine, which was quite an interesting learning moment, and we've been learning ever since on this trial. In this therapy, we engineer patient cells in the following process. The process that we employ at University of Pennsylvania to generate these chimeric antigen receptor engineered T cells is based on a process that was started more than 2 decades ago by Dr. Bruce Levine in Carl June's lab back then. It entails the apheresis of a patient to collect white blood cells. T cells are then enriched using beads that have anti-CD3 and anti-CD28 conjugated to them. This process removes the non-T cells and at the same time activates T cells for transduction with the lentiviral particle that encodes the CAR. The cells are then expanded in a steady-state culture in the first 5 days, transferred to a wave bioreactor, and further expanded until day 9 for the harvest. Beads that have been used to stimulate the selected cells are then removed, and the cells are cryopreserved and formulated at different doses as it was done on this first anti-CD19 CAR T trial. The patients receive chemotherapy for cytoreduction, and then cells are infused in 3 consecutive days at the 10% dose first, followed by the 30% and the 60% at the latest time point, unless adverse events appear. The patients are then followed to track their CAR T cell expansion, as shown on the right-hand side of the slide. TL019 indicates the murine CD19 CAR that we used with different growth curves. And also we monitor the leukemia if it's circulating using a flow cytometry-based assay. But also we used deep sequencing using IGH-seq with a CRO. We next performed also routine CAR flow analyses with an antibody that's specific for the idiotype. And an important part of this translational pipeline was that we collected samples for later research to sort the CAR T cells for various assays such as TCR-seq. Lastly, we employed vector integration site sequence analysis with a collaborator at Penn to understand where exactly the CAR inserted into the genome, which then also functioned as a fate mapping tool for the CARs that were infused. So we treated initially patient with chronic lymphocytic leukemia, which at the time was a high unmet need. There's a background CLL accounts for 25% of newly diagnosed leukemias, the most prominent leukemia in the Western world. It's diagnosed usually at an average age of 70 years of age, and it affects mostly males. Current drug-based therapies that have been developed over the past decade include cell signaling inhibitors such as ibrutinib and others. While these therapies have proven very effective, they come at significant cost, both financially and biologically and clinically, but also they do not induce a cure. Cell-based therapies, on the other hand, are curative, as we have seen with these CAR T cells. The patient, going back to patient number, the first patient reported in our New England Journal of Medicine paper, the CAR T cells were actually quite effective in this patient. His marrow at starting point had a high cellularity, with most of the cells made up of CLL cells. But then by month 1 and also month 3, we could see at the bone marrow biopsy, as well as the CT scans, a dramatic regression of the tumor coinciding with the expansion of the CAR T cells. We followed the CAR T cells using a flow-based analysis like I said before, as well as PCR. This slide shows the PCR in whole blood in figure A, the expansion in the first month with a gradual decline in the ensuing weeks until month 6 is when we follow this patient with this assay. And on the right-hand side in figure B, you can see the expansion curve in the marrow, which was also sampled. So we see that there is a expansion both in the peripheral blood and marrow. That means that the cells will find their tumor targets in the marrow and probably also in the lymph nodes. Analyzing the phenotype of the cells after infusion showed us that the proportion of leukemic cells in figure C that expresses both CD19 on the y-axis and CD5 on the x-axis at baseline was very high, but reduced to barely detectable at 1 month, and at 3 months was no longer detectable by this assay or any other assay that we employed. We measured, aside from the CAR T cell expansion and the tumor response by flow cytometry and sequencing, also the bioactivity of the cells that we infused. And we did that by employing a cytokine multiplexing assay that we have rolled out in all of our studies that measures a set of 30 cytokines in the serum. This slide shows that patient number 2, which was infused with only 14 million CAR T cells, was cured and cured of his cancer, had a dramatic increase in cytokines such as IL-6 and interferon gamma around the time of peak expansion of the CAR T cell. Patient number 1, over 409 number 1 in figure A, showed a much more rapid expansion of the CAR T cells, peaking at day 3, coinciding also with a high rise in inflammatory cytokines. Both patients have remained in remission ever since. Patient number 3, on the other hand, in figure B, subfigure B, only achieved a partial response, and that was matched by a very small increase in inflammatory cytokines after infusion, suggesting that bioactivity was another important biomarker of product activity. And we have learned ever since that some of these cytokines are participants or contributors or drivers even of cytokine release syndrome, and other cytokines actually can help us predict high-grade CRS, such as interferon gamma, which we published with co-workers at Children's Hospital. We follow these patients much longer, for 4 years, as shown in this slide. Again, patients 1 and 2 on the left-hand side achieved a CR. and that was coinciding with an increase and persistence, actually, of the CAR-engineered cells, which were detectable both by flow cytometry, as shown here in the circle symbols, and PCR. Analysis of patient number 1 CAR T cells at the indicated time points in the right-hand side of this figure showed that they displayed activity directly ex vivo. In this experiment, we collected the samples that were frozen in liquid nitrogen, thawed the cells, stimulated for 6 hours to measure intercellular accumulation of cytokines such as interferon gamma and IL-2 on the left-hand side in different colors, and also MIP-1 beta on the right-hand side. And you can see that samples taken from day 50 all the way up to year 3, when stimulated for only 6 hours, were highly active against CD19 expressing target cells. And this, of course, matches with the deep B-cell aplasia in both subjects. To generate a better understanding of the difference between efficacy and failure of the CAR T cells in CLL patients, as we have seen in 2 trials in a combined set of 41 patients, we answered the questions related to the product quality itself, meaning the T cells. The hypothesis being that T cell biology, intrinsic features, play an important role in the clinical efficacy of the drug. We analyzed the infusion product itself and identified pathways associated with T cell exhaustion. The higher bar is really to identify predictive biomarkers in T cells taken before CAR T cell manufacturing. So these are the apheresed cells. Doing this analysis in 41 patients showed us the example as shown on the left-hand side, a phenotype that was made up of CD45RO negative and CD27 positive cells, which was this fraction was much higher in patients achieving complete remission, whereas non-responding patients had a significantly lower proportion of these early memory T cells. We also identified 2 partially responding patient groups, one that had the partial response that was likely mediated by a transient CAR T activity combined with chemotherapy, and a second partially responding patient group that actually transformed into an aggressive lymphoma. And the T cells, CD8 T cells in that group was very similar to the complete responding patients. The discovery cohort showed a significant difference between both CR and PRTD groups compared with the other patient groups. And we then validated this biomarker profile in a 3rd trial of CLL treated with CD19 CARs. So this work led to the development of a predictive biomarker of clinical efficacy of anti-CD19 CAR T cells in CLL, as well as a first identification of cells most likely contributing to the clinical efficacy, and of course leading to process development activities. Since we have infused these patients over a decade ago and we had biobank specimens, we're still analyzing these patients as they come to the clinic because of this gene therapy product. We are also collecting cells, and CLL, we see a relatively high level persistence of the CAR-engineered cells as shown in this slide. So this figure shows on the left-hand side a flow cytometry-based analysis of CD4 on the x-axis versus CAR-19. So the antibody against the CAR detects a high level of CD4 cells expressing this CAR, as well as CD4 negative cells expressing the CAR. We followed patients 1 and 2 for 10 years, like I said, and find that the tumor has disappeared based on a flow-based analysis and sequencing, and the CAR persists throughout, detected by both flow cytometry and by PCR. Because of its high-level persistence and because of the biobank specimens, we could ask additional questions as to the nature of the infused cells. The first question we asked is what the fate of the infused cells was, and we followed these CAR T cells using 2 different approaches. One was based on sequencing the TCR repertoire in CAR-engineered cells, and for that we purified CAR T cells from circulation in these subjects using flow cytometry, which were then sequenced deeply to reveal their TCR repertoire. In that analysis was done in the first 2 years, when we could collect at least 50,000 events from these patients. On the right-hand side of this left side figure, you can see a number of seemingly words. Those are really the highly variable region of the TCR beta chain, which is sort of a clonal biomarker, if you will, or barcode. And then these rectangles on each of these clonotypes shows a different color. If it's white, then that clonotype is not present at that time point. And when it's slightly red or brown red, that means that there is a high proportion of these clones at these indicated time points. So what this figure shows here is in this subject, and 1.8 months and 2.3 months, there were some clonotypes at the bottom side of this figure. They were present at both time points, and some of these clones actually made it all the way up to the latest time point, measured 1.9 years. Whereas others in the top-hand side of this figure shows early appearing clones at 2.3 months that have persisted all the way to 9.3 years. So that's very long persistence of CAR T cells. We also determined CAR T cell persistence by measuring by assessing the lentiviral vector insertion sites. And this analysis was run using a Morosita overlap index, which essentially indicates overlap, high abundance overlap, if there's a large dark blue circle, such as in patient number 2 at month 1.8 and day 31. That repertoire is highly overlapping. And when there's a repertoire that's not over, when there's a clonal composition that doesn't overlap, you see is 0 and no such blue circle. So this analysis run on patient 1 who was infused with 1.1 billion CAR T cells shows that in the first 2 years, there was large clonal turnover, which stabilized at year 2 and has remained stable ever since. Whereas patient number 2 on the right-hand side, there was a stabilization in the first year, followed by a second repertoire taking over and gaining dominance up till the latest time point measured. So we then, because of the biobank specimens, we could also analyze the immune phenotype, and for that we devised a custom-built CyTOF mass cytometry panel based on 40 markers, and we analyzed the specimens of 5 time points for each patient, 10 time points in total, 40 markers. And in this analysis, we included a metal conjugated antibody against the CAR. In total, 45,000 cells were collected across all time points. In patient 1, we saw a high prominence of CD8 positive CAR T cells at early time points, which diminished over time and was dominated at later time points by CD4 T cells. In the CD4 compartment, we identified cells that were highly proliferative in dark blue, expressing Ki67. You can see that in patient number 1, this persisting CAR T cell population was indeed proliferating substantially. Whereas the second patient, we see that there is a population of CD8 positive, but also CD8 negative CAR T cells. Later analysis revealed that the CD4 negative, CD8 negative population was a gamma delta T cell population. And as in patient number 1, this population diminished over time as well and was dominated by highly proliferative CD4 CAR T cells. We next mapped some of this phenotype using, again, the UMAP, which is a dimensionality reduction tool. On the left-hand side, you can see the 5 populations as previously described. In brown, we see the CD4 negative, CD8 negative gamma delta population. In the middle, in red and in yellow, those are the CD8 cells. In green, CD4 positive population that actually dominated at later time points. Our next analysis revealed we did by single-cell RNA sequencing combined with immune phenotype assessment with antibodies, as well as 5' TCR site sequencing. This analysis was done on purified CAR-expressing cells at 9.3 years after infusion. We collected a total of 1,437 sorted cells. Most of those were CAR T cells, and we also sorted a CAR-negative population as a control. The left-hand UMAP reveals these different populations. You can see that the transcriptome of CAR-engineered cells on the right-hand side, sort of the foot-shaped populations, are very distinct from the left-hand side, the CAR non-expressing cells, which are largely quiescent. We see a predominance of both 4s and 8s on the CAR-negative cells, whereas the CD-CAR-positive cells are largely CD4 positive. Analyzing these UMAPs in figures B through E revealed that indeed the CAR-positive cells were on the right-hand side. The clonotypal composition in C shows that this population is dominated by a single clone with some smaller subclones, and CD4 protein assessment was done, as well as CD8 analysis. Importantly, what this study revealed is that the long-term persisting CD4 positive CAR-engineered cells were highly activated, and we know that because we analyzed the cell cycle signatures of the S phase, as shown in figure F. S phase cells on the right-hand side bottom, the tip of the UMAP, whereas the G to M cells, also cycling cells on the right-hand side. The purple-blue domain is mostly expressing cytolytic molecules. Furthermore, we revealed that the activation markers CD38 and HLA-DR were highly expressed on all of these CAR T cells, as well as immune checkpoint regulators such as PD-1 and TIGIT, TIM3 and LAG3. So this led us to conclude that this long-term persisting CAR T cell population was highly activated, but with a restrained activation signature. We furthermore found that these cells were highly metabolically active, which you can find in the paper that we published on this. But also, since these persisting CAR T cells expressed signatures of cytolytic activity, such as perforin on the top left-hand side of these UMAPs, as well as coenzymes A and K, suggesting that these CAR T cells were actually able to kill target cells. And that we confirmed in a killing assay using CD19 engineered cells, as shown on the bottom of this figure. So in essence, what we have shown in these two subjects, actually mostly in subject number 2, I would say, is that the memory-centric CAR design, so including a BB-zeta domain, has endowed patient T cells to target their own blood cancers, with the 4-1BB vastly outperforming in long-term persistence compared with CD28. And that's also confirmed in previous studies by Crystal Mako's lab and others. Correlative studies have fueled a steady stream of hypotheses and experiments, and because of the biobanking, we were able actually to perform some mechanistic studies and find important biomarkers, but also novel pathways for cell engineering. We also identified mechanisms of toxicity, such as cytokine release syndrome, which has allowed us to develop a predictive biomarker profile. The persisting CAR T cells remain functional. Initially, we showed that for up to 3 years, but now also at 9.3 years. Taking these cells directly from patients showed a highly functional profile. And also, we see that CD4 positive CAR T cells are actually the dominant subset, which sustain remission of leukemia. And the population exhibits a restrained activation state. Possibly that's an important feature of long-term persisting memory T cells. One question one may want to ask is CD19 the best possible target antigen? The fact that one of these two patients that was treated with CAR-19 died of COVID, likely because of lack of B cells, suggests that we do really need to find better ways of targeting leukemias. But anyway, with this first proof of concept, if you will, CAR T cell therapy, we have shown that the therapy can actually induce a cure of leukemia. This work was done by a very large team, as shown on this slide. The top left corner is my team as it was at Penn. I'm now at Cleveland Clinic and building a new team and have open positions for all levels. I'm very thankful also for the patients that were participating in these early phase trials, as well as clinicians such as David Porter, of course, Dr. Jun, and Dr. Grubb, and the folks in my group and the Children's Hospital. And I thank you very much for your attention, and I'll take questions if you have.